Good evening, everyone, and thank you for joining this educational webinar. My name is Dr. Alka Goyal. I'm the Director of Laboratories at Genesis, and today I will be speaking to you about egg freezing from the laboratory perspective. Most discussions about egg freezing focus on the clinical side, hormones, stimulation, and decision making. Today, I'd like to take you inside the lab because once the eggs are retrieved, the lab plays a very critical role in protecting them and preserving their potential. This session is not meant to convince you to freeze or not to freeze your eggs. My goal is to give you a clear understanding of the science behind this process so that you can make an informed decision together with your physician. I'm going to walk you through what happens to the eggs step by step from the moment they arrive into the lab through freezing, storage, and eventually thawing when they are being used. I'm going to try to keep this as simple as possible. I promise there's going to be no exam at the end. Egg freezing, scientifically called oocyte cryopreservation, is a fertility preservation technique that allows women to store their eggs for potential use in the future. Now, women choose egg freezing for various reasons. Some are focusing on career goals, some may not have the right partner, others may be preserving fertility for medical, personal reasons. From a scientific standpoint, egg freezing allows us to preserve the egg at a biological age that it was frozen. Before we go into the lab, I want to briefly talk about why the timing of egg freezing is considered a very critical decision in preserving fertility. Egg freezing is important because it's closely tied to how human biology works. The most significant factor affecting fertility is age. As age increases, egg quality declines, particularly at the chromosomal level. In the later ages, even though eggs may be present in the ovary, their developmental potential may not be the same as this changes over time. So, egg freezing allows us to preserve the eggs at an earlier biological age. In that sense, it gives us the ability to pause time for the egg, even though time continues for the individual. However, it does not guarantee a future pregnancy, and neither does it improve the quality of the eggs. It simply preserves them as they are at the moment. Future outcomes depend on several steps after they are thawed, which will be discussed later. So, when someone considers egg freezing, they are not making a decision just about a single event. They're making a decision about a process that involves multiple steps. And the understanding of this process is what is going to set realistic expectations. Now we know that egg quality is the most important factor in reproductive success. Let's understand what are the factors that affect the quality of the egg. As we've discussed, the age at the time of freezing. Now, why does age matter so much? Because age directly affects chromosomal integrity. And as age increases, the chances of abnormalities in these chromosomal parts definitely increases. Second is the natural biological variability. Not all eggs, even from the same cycle, are considered identical. Some have higher developmental potential than others. Third, the ovarian response, which reflects the biological environment in which the eggs are developing. Once eggs enter the lab, additional factors come into play: egg maturity, lab conditions, subsequently survival after thawing. And beyond the lab, success depends on fertilization, embryo development, implantation. So egg quality is just the starting point, but the journey to pregnancy involves multiple biological steps. Egg freezing does not improve the quality of the egg, it just preserves it. Finally, we go into the lab. Before we go into the lab, a quick overview of what really happens clinically in order to collect eggs. The ovaries are stimulated for about 10 days to produce multiple eggs. This is monitored through ultrasound and blood tests, which is followed by an egg retrieval, which is a minimally invasive process whereby fluid is collected from the follicles. And this is the point where the lab takes over. During retrieval, the physician aspirates follicular fluid from follicles, which is collected and immediately brought to the embryology lab. As you can see in the picture here, an embryologist is working and scanning the fluid under the microscope, looking for eggs. Each egg is collected and placed in specialized culture conditions. From the very beginning, each egg is carefully labeled and tracked. we use a strict chain of custody system whereby two embryologists verify and document each step to ensure that there are no mix-ups. The eggs are then placed in incubators, which mimic the body's environment, maintaining the stable temperature, pH, and protective conditions. This is what an egg looks like under a high-powered microscope. An egg actually is the largest cell in the human body, but even then it's only 1/10 the millimeter in diameter. When we examine drops of fluid under the microscope looking for eggs, these are almost invisible to the naked eye. Sometimes it's like looking for a contact lens in a bottle of water. Once identified, the next step is assessment. Eggs are initially surrounded by support cells, which are called cumulus cells. These cells help nourish and protect the eggs while it develops in the ovary. In the laboratory, we gently remove these cells so that we can clearly visualize the egg itself. And we need to do that because we have to determine the maturity of the egg. This step is very important because only mature eggs are suitable for freezing and then later on for fertilization. This picture is showing you eggs before and after the removal of the cumulus. On the left, you can see the egg is got cumulus on it, and on the right, it's a clear picture of an egg. And this allows us to look at the egg from the inside, and we are able to assess the maturity of the egg. Now, what are mature eggs? Eggs are classified into stages. The stage that we are most interested in is an M2 or the metaphase 2 eggs. These are called mature eggs. They have completed the necessary steps required for fertilization and contain the correct number of chromosomes. Immature eggs, that is M1 and germinal vesicles, are not suitable for freezing as they are not ready for fertilization and they do not have the right number of chromosomes. Because of this, only mature eggs are chosen to be frozen. This is a picture on the left is a mature egg, as you can see, and on the right, there are two immature eggs. The one which has got a vacuole kind of structure in it is called a germinal vesicle. One important point to remember is that every egg is biologically unique. Even as eggs from the same cycle, they can behave very differently. Some eggs are stronger, some are fragile. The variability is natural. This is a slide showing you different kinds of eggs. As you can see, there are all sorts of things inside and outside the egg. These are variability of eggs. This is a slide showing you different kinds of eggs. But when we see some of the differences, what we can visually see them, but some of the differences are biological and we cannot see them. The variability is a part of natural biology. Freezing does not create these differences. It simply preserves the egg as is. This is one of the reasons why doctors often recommend freezing multiple eggs, because biology always involves some sort of natural variability. Now that we have selected mature eggs, we can move to freezing. The method that we use for freezing is called vitrification. This is a rapid freezing method which prevents the formation of ice crystals. Eggs, like the rest of the human body, is made up of approximately 80% water. If frozen slowly, this water is going to turn into ice crystals and it will damage the delicate structures of the egg. With rapid freezing, the water turns into a glass-like state instead of ice. That is what protects the eggs from its internal structures. To prepare the egg to withstand these very low temperatures, sometimes water from the egg is replaced with protective solutions. These are called cryoprotectants. Then the eggs are rapidly cooled on specialized devices. This process must be carefully and precisely done so as to protect the egg. You can see in the picture here, the picture on the left is showing eggs with ice crystallization. These can actually damage the internal structure and the eggs can degrade. And on the picture on the right is showing the egg being transitioned into a glass-like stage, which is the hallmark of vitrification. These are the various stages. As you can see, some of the eggs are getting dehydrated. We take some of the water out. We expose it to cryoprotectants. These are just the stages in freezing. This is an embryologist preparing and placing the eggs into specialized devices to be rapidly cooled. Each step is performed under a microscope because eggs are not visible to the naked eye. Each egg is individually labeled and a chain of custody is very much followed here. If you can see the devices that we actually store them in are called HSV straws, and these are then put in canes and then they are directly put into liquid nitrogen. This rapid cooling is what prevents the ice crystals from forming, which protects the eggs. That is why modern freezing works much better than the older freezing methods. After freezing, the eggs are stored in tanks which are containing liquid nitrogen. The temperature inside these tanks is approximately minus 196 degrees Celsius. That is approximately minus 320 degrees Fahrenheit. At this temperature, all biological activity stops. This means that the eggs do not age while they are frozen. Eggs can remain safely stored there for many years in these conditions. I think the longest time that the eggs have been frozen and taken out for use has been nearly 14 years. Embryos have a larger lifespan on that, so I think that is around 30 years. So this is one of the few times in biology, actually, if nothing happens, it's a really good thing. When you're ready to thaw the eggs, they are thawed in the same way in a very careful stepwise process, very similar to freezing. After that, we assess survival, those that are now ready to be fertilized. If the ones which have survived are now ready to be fertilized. Freezing preserves the egg at a certain age, but success still depends on how the eggs are going to perform. Not all eggs survive freezing and thawing. Some of the loss is expected, some is biological variability. This is not failure, which brings us to the question, why do all eggs not survive and become into embryos after fertilization? Eggs are very specialized and delicate cells. Because of their size and complexity, they are sensitive to stress. The internal structure, especially the spindle that organizes the chromosomes, is particularly sensitive to stress. With modern vitrification, about 90% of eggs survive, which is a very good outcome. An easy way actually to think about eggs is to imagine seeds in a garden. Not every seed planted grows into a plant. Some germinate, some do not, some are stronger than others. Eggs behave in a very similar way. Each egg has a potential, but not every egg is going to develop into an embryo or lead to a pregnancy. The lab environment actually plays a very critical role in successful freezing and thawing of survival too. It requires extremely stable conditions. High quality labs maintain extremely high and very good stable conditions. Temperature, air quality, and culture systems are all standardized. They have rigorous quality control over all their systems. The technology that they use is advanced. Equipment, freezing protocols, all undergo a rigorous quality control, and all are essential for the optimum thawing of eggs. Equally important is the experience of embryologist performing these procedures. So the next logical question becomes, how many eggs are needed? How many eggs should one freeze? For that, we have to understand what really happens to the egg after it is thawed for use. An egg doesn't directly become into a pregnancy. It has to go through several biological steps. After thawing, 90% of eggs survive with modern vitrification. Of those, roughly 70 to 80% of them will fertilize. From there, only 30 to 50% will develop into blastocysts. These are the embryos which are suitable for transfer. And then all embryos are not chromosomally normal, especially as age increases. And finally, even a normal embryo must successfully implant into the uterus. So what you can see here is that at every step, there is a natural biological attrition. That's why one egg does not equal to one baby. And this is why when we talk about egg numbers, what we are talking about is probability, not certainty. Because of this stepwise attrition, physicians often recommend freezing multiple eggs and sometimes freezing multiple cycles. Younger individuals may need fewer eggs, while age increases, more eggs are needed to achieve the same probability. The estimates shown here are based on predictive models from large IVF data sets and represent averages. Individual outcomes vary significantly. Now, as you can see here, under the age of 35, maybe 15 to 20 eggs are enough to give you a 70 to 80% chance of more than one live birth. But keep in mind that these are just guidelines. These are not guarantees. These numbers are not targets. They are probability estimates based on population data. There are several concerns that people really can have about egg freezing. So one of the major concerns is do my eggs lose quality over time? At ultra low temperatures, eggs do not age. Do they get damaged? Egg damage during freezing is very minimal because with modern vitrification, survival rates are nearly 90%. Then another thing which a lot of people ask is frozen eggs weaker than fresh eggs? They they certainly are not, because once they thaw out and survive and they go through fertilization, with modern vitrification techniques, fertilization and pregnancy rates are comparable to that seen with fresh eggs. Some people also ask if egg freezing is an experimental technique, but the American Society of Reproductive Medicine, that is the ASRM, declared it no longer experimental in 2012. IVF laboratories use strict identification and tracking systems to prevent any mix-up of eggs. This is just an idea to give you what egg freezing can do and what it cannot do. Egg freezing is a powerful tool, but it's sometimes important to understand both its capabilities and its limitations. Egg freezing can preserve your eggs at the biological age. It can provide future reproductive options. It preserves options, peace of mind. But it cannot guarantee a pregnancy. It cannot improve egg quality, and it cannot remove all uncertainty. So the key takeaways from this would be that egg freezing certainly is a powerful fertility preservation tool. The lab plays a very critical role in protecting each egg, and understanding the science will help you make informed decisions. Egg freezing is not about predicting the future. What science can do is preserve possibilities. It does not guarantee a baby, but of course, it offers something meaningful: time and options. If you remember one thing, remember that egg freezing allows us to pause biological time for the egg, even though it continues for us. Thank you very much, and I would be glad to take your questions. So in the natural cycle, remember that only one egg matures out of, we will just be left with just one egg. So what the medication does is actually starts the development of lots of eggs, because we are now interested in numbers. The more the eggs we have, the more of them will survive, the more they will fertilize, and perhaps we'll have more embryos, because all eggs are not exactly the same. So what the medication does is improve the number of eggs that are going to mature. So in the natural cycle, 5 or 6 eggs are going to start maturing, then only one of them is going to become dominant, and that's going to be the egg that is, going to be released, and then it'll go for fertilization. When we take medication, instead of one egg being dominant, there are 5, 7 eggs or maybe 10 eggs that start growing, and we are able to now then remove them, and they are mature eggs. That actually is a clinical question which could be answered by the physicians. That depends. It's very individualistic. The doctor will decide, see what your ovaries look like, whether you're doing well, and it's a clinical question. So I would suggest you could ask this to your doctor, but basically it will be decided by a physician. So, we are born with more than 30,000 eggs in each ovary. So each of those 30,000 do not have the same potential. So I always like to give the analogy of seeds that, we have many seeds, but all of them do not germinate. All of them do not, they do have the potential to become a plant, but all of them don't have the strength. So that's why we like them in numbers. No, that shot that you're talking about, your trigger shot, they might have changed the trigger shot. It's got nothing to do with with your abnormality or normality of the embryo. Keep in mind, this is again completely unconnected with age. Now, if you think of an egg, all the cells in your body, I'll just go into a little bit of science so that you understand what normality and abnormality really is. That if you look at any cell in the body, it's got 46 chromosomes. But an egg does not have 46 chromosomes simply because it has to have half the number of chromosomes. The other half has to come from the sperm. Otherwise, every subsequent generation would have, how many ever 46 plus 46 chromosomes. So the egg goes through a maturation process, which is called meiosis. That's why we talked about metaphase 2 eggs, which are mature egg, in which that it reduces the number of chromosomes into half. That is the process of maturation that we're looking for. Sometimes that as you grow older, instead of half, exactly 23, it makes a mistake at the time of maturation. It has 24 or it'll have 22. So when the sperm comes in and it fertilizes with the sperm, which is, let's assume, has got 23, the resultant embryo sometimes will not have 46 chromosomes. It'll have 47, or maybe it'll have 45. And that's what is an abnormal embryo. So it's actually got to do completely and totally with the egg, or sometimes even the sperm, because the sperm also goes through the maturation process. But keep in mind, 90% of the machinery that goes into making a human being is in the egg itself. So the egg is always the usual suspect. So remember, I have to bring you back to the seeds analogy again. When you are doing, if in the one cycle, let's assume there's a whole sack full of seeds over there, you're picking up a fistful of seeds. The chances are that the good egg is in there. It's every cycle is different. And just because you have abnormal embryos in one cycle doesn't mean you're going to have it in the other. Your abnormality or normality of the eggs and the proportion of those is directly related to your age, because as eggs age, they are unable to mature successfully, and that's why sometimes there is a difference in chromosomal numbers, and that's why there is an abnormality. The medication is just increasing the number of your eggs. I think we don't know that as yet. There's no such thing that if I tell you that you have CoQ10 or have vitamin C or something like that, I think the most important thing is to improve your general health. And of course, keep the stress factor out of it. Hard as it is, stress plays an important role in everything. No, in eggs, I do not know whether eggs are abnormal or normal because we are not doing any genetic testing on them. I will freeze all mature eggs. But yes, the embryos, which turn out to be abnormal, we give you an option. We do not throw out any embryo. We will ask you for a consent. You're going to consent that this embryo is not being used, it's abnormal, and then we will throw it out. But eggs, we do not know the chromosomal status of the eggs because we do not do genetic testing on the eggs. No, it's not automatically discarded because once an embryo is created, it becomes your property. So we would not legally throw out an embryo unless you consent to. So whether you want to keep the abnormal, some people keep the abnormal embryos because for different reasons. and they just don't want to throw it away till they finish all the family planning. And some people decide to throw it out because they are never going to use them. They are never going to risk transferring them because they don't want to risk ever having an unhealthy child. Some people actually are willing to take that risk. No, Down syndrome is a condition that is of an embryo. An egg does not, an egg will not have a Down syndrome because, as I told you, the egg has got 23 chromosomes, right? And Down syndrome is 47 chromosomes. So the other 23 comes from the sperm. So once an embryo is created, then we know the exact number of chromosomes.